Hi, my name is Vijay Kumar, and in this video we will talk about genetic circuits and how they can be used to cure and detect cancer. How does the cell work? A cell performs computation all the time. It senses the information, makes some decisions, it stores the information and gives output according to the information processed or the computation performed. What are genetic circuits? Cells can be programmed to perform a particular logical function with the help of genetic circuits. The behavior of a cell can be controlled using genetic circuit, which performs computational signal processing and communication. Since the field of synthetic genetic circuit is still in the infant stage, there is a lot of development that has to be performed. The basic logical function that can be simulated by a genetic circuits are NOT gate, NAND gate, and implies gate and AND gate. What is cancer? Cancer is a disease that involves abnormal growth of the cells in the human body. These cells can invade other tissue organs. Is cancer a genetic disease? Yes, cancer is a genetic disease, but it is not necessarily a hereditary one. However, a child can inherit some genes from the parents which might increase the probability of development of cancer in the child to some extent. It starts with a single hyperproliferative cell which then grows and divides into a small adenoma, which on further development becomes a carcinoma or a large tumor cell. Now, how to cure cancer? Nowadays, most of the cancers can be treated using various techniques available in the market, like surgical methods, chemotherapy, and radiotherapy. A lot of research has been going on to find a cure for cancer at gene level. How genetic circuits can cure cancer? Now, with the help of genetic circuit, we can give the human cell ability to sense and detect disease markers by encoding conditional production of a molecule which fight back the disease when it is sensed by our circuit. Now, hypoxia inducible factor 1, HIF1, is a protein which helps the cell to grow in a low oxygen environment. Cancer cell grows very rapidly in the body and they form a large mass. This large mass puts pressure and compression on the nearby blood vessel, causing the low inflow of oxygen to the center of the tumor. In such a condition, the cancer increases the production of HIF1 protein, which then restabilize the metabolism of the cell and send signal for the construction of new blood vessel around the cancer mass. As we can see in the figure, the genetic circuit used act as a logical AND gate. In our case, when the cells are in a state of hypoxia and when we have added a doxycycline compound to the body, then only the output of the AND gate is true. So when the output of the AND gate is true, the circuit promotes the production of a molecule called cyclic hexapeptide using Cyclops method, which has been experimentally determined to inhibit the HIF1 protein, thus preventing the further growth of the tumor in the body. Genetic circuits for the early detection of lung cancer. Detection of cancer using conventional methods require high cost and is a long process, whereas early stage detection and further treatment of cancer using tumor marker has gained a lot of popularity because of its efficiency. Markers that can be used for detection and treatment of lung cancer are CEA and CYFR21. The genetic circuit for the early detection and further treatment is composed of two parts: single marker detection circuit and an integration circuit which computes all output produced by single marker circuit. The main component used in single marker circuit as well as integration circuit is the circuit implementing the electrical NOT gate. Now in single marker detection circuit, Inverter circuit is mainly used to detect the high concentration of one of the three markers. The input module in the single marker circuit has the responsibility to detect input signal continuously and pass on the information forward. rRNA's input concentration can influence the output signal of the single marker detection circuit and threshold and can destabilize the circuit. Hence, high concentration of RNA is not desired. Integration and gate circuit. The integration circuit is necessary to construct a multi-index circuit network to deduce the output data from a single marker detection circuit using logical operations. If the integration circuit receives an input from a single marker circuit, the genetic circuit in the cell will be activated with green fluorescent protein GFP produced in order to provide an early warning of lung cancer. In our circuit, logical AND gate is used and is implemented using logical NAND gate followed by an inverter circuit. Hence, we can see that GFP will be activated only when both the inputs are present. The full detection circuit. As we can see in the figure, the full detection circuit of lung cancer consists of each single marker detection circuit along with the AND gate integration circuit. The aim of single marker circuit is the concentration test, whereas the aim of integration circuit is to apply logical operation on the input received. According to the research, when TPS is greater than 80 UL and CYFR21 is greater than 4 nanogram per ml and CEA is greater than 5 nanogram per ml is taken as a threshold value, high sensitivity and specificity can be achieved.